The cell cycle is a necessary and continuous process by which a cell divides, differentiates, or dies. Cell division is necessary for single-celled organisms to reproduce, and for multicellular organisms to repair damaged cells and grow. The cell cycle begins with interphase. During the first or G1 phase of interphase, the cell increases in size and synthesizes organelles and proteins. Before moving on to the S phase, the cell checks to make sure it has sufficient resources and is healthy. The next phase of interphase, called the S phase, is very important. During this phase, the cell replicates its DNA in the nucleus of the cell and duplicates its centrosome organelle, necessary for cell division. The final phase of interphase is the G2 phase, during which the cell's organelles double, a new cytoplasm is formed, and additional structures necessary for mitosis form. The next phase of the cell cycle is mitosis, which is broken into 4 subphases. Compared to interphase, mitosis happens relatively quickly, especially considering how many steps are involved. The first phase of mitosis is prophase. Chromosomes condense, the nuclear membrane disappears, and a spindle forms, anchored by centrosomes at opposite poles of the cell. Next is metaphase. The chromosomes line up on the cell's equator, and the spindle fibers attach to this part of the duplicated chromosomes called a centromere. After metaphase is anaphase. The 2 sister chromatids of each pair separate, and the centromeres divide. Then the chromatids move to opposite poles of the cell. Finally, the cell enters telophase. New nuclear membranes form around the chromosomes and they uncoil. The uncoiled chromosomes are called chromatin. The final phase of the cell cycle is cytokinesis, during which the cytoplasm divides to form 2 identical daughter cells. After cytokinesis, cells re-enter the G1 phase. Sometimes a cell will exit the cycle near the end of this phase. This can occur when a cell differentiates or decides to stop dividing to specialize on a task within an organism. A cell might also exit the cycle if it has insufficient resources to continue growing and dividing. Some cells, like neurons and muscle cells, will remain in the non-dividing or G0 stage. However, damage or injury may prompt cells to re-enter the cell cycle. The cell cycle is by no means a perfect system, and sometimes mutations in the cell, specifically in the cell cycle regulators, can cause problems. Cell cycle regulators are proteins that either stimulate or inhibit cell cycle progression. Stimulating proteins are encoded by proto-oncogenes, and inhibiting proteins are encoded by tumor suppressor genes. Checkpoints during each phase of the cell cycle make sure the steps are growing properly. The regulator proteins can halt the cycle to fix errors or stimulate it to progress. However, if mutations occur in either the proto-oncogenes or tumor suppressor genes and the cycle is improperly regulated, too much cell division or too little cell death can form cancerous tumors. Understanding these cell cycle regulators is important to the development of cancer treatments and to understanding the cell cycle as a whole.